Hello, everybody. Welcome to Ms. McGuire online video lecture, and today's lecture is about changing allele frequencies. Learning outcomes for these chapters. Students will be able to explain how non-random mating changes allele frequencies in populations, explain how migration changes allele frequencies in populations, explain how the random fluctuations of genetic drift affect genetic diversity, discuss how founder effects and population bottlenecks amplify genetic drift, discuss how mutation affects population genetic structure, provide example of negative, positive, and artificial selection, and explain how balanced polymorphism maintains certain diseases in populations. In the previous chapter, we talked about Hardy-Weinberg equilibrium. Now, this equilibrium tells us that if we have large population, then allele frequencies have tendency to stay constant. So it's about unchanging allele frequencies from generation to generation. So, for example, just to make it understandable, let's say you're doing research on a population of rabbits. And let's say it's a huge population, 100,000 rabbits that live in a particular area. So you go 1 year to study population and you notice that, well, 60% of rabbits are white, 20% of rabbits are gray, and 20% of rabbits are, black, right? So that rabbits are white, gray, or black, that's determined by their genes and alleles, right? Because alleles are different variation of genes. So let's say 10 years later, you go back and you study the same population, and Hardy-Weinberg principle tells you that you still should see 60% white, 20% gray, and 20% black rabbits, right? So that means population really, allele frequencies stay constant in a large population. However, that's not always true, right? So what if you return back 20 years later, and now you found that instead of 60%, you have 70% of white rabbits. That means that allele frequency changed. Now, what are those conditions that can change allele frequency? And ultimately, phenotypic frequencies, right? If we change allele frequency, then we change phenotypic frequencies of those rabbits. Now, instead of 60%, maybe you have, doesn't necessarily need to be 70, maybe it's just 61%. But those 5 conditions that change allele frequency are non-random mating, migration, genetic drift, mutation, and natural selection. So over here, this show you those conditions that can change allele frequencies in a diagram. Right, so here our population, and let's say we have, I don't know, some animals. Some are blue, some are green, yellow and red, and we have 25% of each, right? 25% of green, 25 blue, and so on, right? But what if blue mates more? So more, I don't know, maybe it's some alien population, alien rabbits. So blue rabbits, they reproduce more often than what do you expect? You expect their phenotype gonna be increased in percentages, right? Or that means allele frequency of blue rabbits now bigger than 25%, right? So that's what can happen if, blue rabbits have some advantage and they just reproduce more often. Also, we can say, like, what if red and yellow will join the group? So more rabbits, alien rabbits, right? Because we don't have blue, green rabbits, but it just you know, it just analogy. So what if red and yellow enter the population? Well, then you have more of yellow and more red, right? Or genetic drift of the green and yellow. So let's say by the chance, because genetic drift means it just, you know, for example, it was some disaster and accidentally blue and red were killed. And then we have more green and yellow. Or it can be some mutation and those blue became red. So now we have less blue, more red. Or it can be natural selection. Now, natural selection, when green cannot reproduce, for example, then we don't have green at all. And we now have different allele frequency because of natural selection. Right, all these are actually kind of random and only natural selection. It's a result of adaptations. Okay. So let's now talk about these 5 factors in more detail. So the first is non-random mating. Indicates that individuals of one genotype reproduce more often with each other. This is a major factor in changing allele frequencies in human population. Traits that influence our human mate choice include physical appearance, ethnic or religious preferences, intelligence, and shared interest, right? So we don't have random mating. Random mating would be you just go outside and you're like, okay, let me marry this guy or let me marry this girl. And I don't know, it's just the first person I met and we're gonna have kids together, right? So of course we do not choose our spouses this way. We do have non-random mating, right? And it's changing our population. Hopefully we all choose intelligent people to marry and we will have more intelligent people in our population. Oops, I'm sorry. I just wanna. So examples of non-random rating includes males that fathering many children. For example, in Hopi Indians, I'm not sure. Oh, sorry. So they, because one father was albino, because he had many children, then we have increase of allele for albinism in that population. Or Genghis Khan in Asia, 16 million men living today share his Y chromosome. You can imagine, right, how many kids he had. Avoidance of genetic disease with controlled mate choice and reproduction. That's also non-random mating. For example, in some Jews population, they have high frequency allele for Tay-Sachs disease. So if you know if two people know that Tay-Sachs disease runs in their families, they might choose either not get married or maybe even if they marry, they might choose not to have kids, right? So that's avoidance of genetic disease with control mate choice. Consanguinity and endogamy also contribute to non-random mating. So consanguinity is a marriage between blood relatives that increases the proportion of homozygous in the population, and endogamy is a marriage within a community. So both of these are examples of non-random mating. All right, so this is pretty much, just to put it simple, this, for example, if we live in a community because of some religion preferences and males and females, like women and men, they only choose to get married within their community, right? And of course, some might choose to marry their second cousin or third cousin. That's a marriage between blood relatives. Okay, another factor that affects allele frequency and changing allele frequency is migration. So large cities with their pockets of ethnicity defy Hartley equilibrium by their very existence. Individuals migrate and move genes from one area to another. The addition or removal of alleles will alter the genetic frequencies. Migration can result in admixture, which is the introduction of new gene variant into the population, into one population from another. This is followed by reproduction between newcomers and residents. So what it means, like, of course, if we have more people coming to our cities or our villages or our communities, those people bring their own alleles into our population. And for example, let's say we live, people live somewhere and there is no, in the community, we don't have people with green eyes. And then 5 families came with many kids, and all kids have green eyes. And then those kids get older and they marry, found spouses who lived there before. So they came to this population with different alleles, with alleles for green eyes. So they brought their genes within that population. So we expect more kids now be born with green eyes possibly, right? So that's, again, every time we change allele frequency, right, then we are not in equilibrium. And that actually means that population is changing and population is evolving. A genetic effect of migration are reflected in current populations. In a situation termed a cline, allele frequencies change from one neighboring population to another. A cline develops as immigrants introduce alleles and immigrants remove them. Clines may be gradual if nothing blocks migration path, or abrupt if barriers block gene flow. Barriers include geography and language, right? So obviously, when immigration, when we enter population or when we leave a population, as I already said, we take our alleles with us, right? And that's what can be, that's what is called cline when population allele frequencies changes. Another factor, so it does make sense, right? If we decide to marry somebody with a specific traits, right? If all women on this planet decide to marry guys with I don't know, like red hair, for example. So we're only gonna marry from now on guys with red hair. So what would you expect then? Then we would have more red-haired people in the population, right? That's non-random mating. Now, if we move from one population to another population, so they're also changing that population allele frequencies, right? So that's, in this chapter, we're talking about changing allele frequencies. Another factor that changes allele frequency is genetic drift. It refers to fluctuation in allele frequencies from generation to generation that happens by chance to gametes. It is a characteristic of all population. Allele frequency changes that cause genetic drift occur at random and are unpredictable. Effects are accelerated when population become very small and sampling changes allele frequencies. So genetic drift just means that something happened randomly that change a population. So for example, let's say we have population of cockroaches, and I don't know, 80% of those cockroaches are brown and 20% of those cockroaches are red. And all of a sudden at night, I wake up and I just step on, I don't know, let's say it's a small population. Let's say I have only 10 cockroaches. Yeah, that's a horrible example, but I hope it will make sense. So let's say in my house, I hope it will never happen, but let's say I have cockroaches that live in my house, only 10 of them. And let's put it this way, 5 brown and 5 black. So at night, I wake up and I, accidentally stepped on cockroaches and killed 3 of them, and all of them happened to be brown, just by chance. Well, now when these cockroaches reproduce, I will have more black cockroaches than brown cockroaches, right? Because I still have black left and only 2 brown. So now if black reproduce, 5 black reproduce, and 2 brown reproduce, so in a couple years, I expect to have more black cockroaches, right? Unless I will just call somebody to take care of my house and get rid of all those cockroaches. But nevertheless, that's what genetic drift is, when population changes because of chance. And especially when population is very small, right? And yeah, then it has a greater effect. Effects that create small population enhance the effect of genetic drift, right? Migration. So if you have small population, why? Because of migration. Then we will enhance the effect of genetic drift, or natural disaster, or geographic barrier that isolate small groups, the consequences of human behavior. 2 factors that can accelerate genetic drift are founder effect and a population bottleneck, which both decrease population size. So again, just to summarize, genetic drift when we have change in allele frequencies because of chance. Now, how much population are affected by genetic drift depends on how big a population is. If I have 1 million cockroaches, right, and I kill 3 brown cockroaches, it's not going to change population much. But if I have only 10 cockroaches and I kill 3, well, that's a big number. So now what make population small? Here there is 2 factors, founder effect and population bottleneck. So let's see what it is. So here's our genetic drift and 2 types, founder effect and bottleneck. So here's our original population. And you can see we have different alleles. We have blue and red and green and yellow, right? And then, something happens, some of this, let's say these individuals, they pretty much move to a new area, new island, and they started new population. Now, what this population is the same population, not the same, but look, you had 2 green. So, and 1 blue, 1 red, and 1 yellow. Well, so this green, when they start reproducing, then you expect to have more green alleles than any other alleles. And compared to original population, in this population, allele frequency is different, right? So let's say yellow, 1, 2, 3, 4. So it's a 4 over 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 4 over 12, or here yellow, when here our yellow is 1, 2, 3, 4, 5, 6, 7, 8 over, so yeah, I'm not going to count all of this, but just because you started with 2 green originally, right, now you have more green allele in your population compared in proportion to other alleles compared to original population. So founder effect, founder tells you, well, we're gonna establish a new population. So we're just gonna move on some island and start new population there. Bottleneck effect is like when population, so again, this is a lot of genetic diversity within this original population. But when some disaster happened, all of them dead and only some survived. And if it happens that more green survived by chance, then we will increase frequency of green allele in the population. But the whole point over here is, doesn't matter how you look at it, right? The new population now is smaller, right? So now population is smaller. And now a new population might be different from original population. Now, any changes here in a new population will be more drastic compared to original population. So if you for example, kill, one red over here, you only left with one red. When here you kill one red, you still have those red individuals within the population. Right? So the smaller is population, the the more effect on the population has genetic drift. So founder effect occurs when a small group leaves home to found new settlements. The new colony may have different allele frequencies than the original population. It may by chance either lack some alleles or have high frequency of others. Geneticists recognize a founder effect in a community known from local history to have descended from a few founders who brought in inherited traits and illnesses that are rare elsewhere. So, for example, over here, we can see that's a founder population in Costa Rica. So only 4,000 people established this population of Costa Rica. Can you believe it? Like only 4,000 people came to Costa Rica, and now it's 2.5 million. But they're all descendants of these 4,000. Oh, look, over here, or 80. Arteritis. I don't know who these people are, sorry, but it's only 80 people started the population. Japan, oh my goodness, only 1,000, and now it's 120 million. And so that's a founder population, right? When a small group of people move to new area, and now after so many generations, we have many of them. In the Quebec, I know that one in Canada. A founder effect is easiest to trace when historical or genealogical records are available. Afrikaners, for example, Afrikaners of Southern Africa, another type of evidence. So that's the people, I guess it's people from Europe who came and live in Southern Africa. So we know, we just have a historical records of them going to that place. Another type of evidence is when all individuals in the population with a certain illness have same mutation. People in Puerto Rican ancestry in East Harlem, they have this Steele syndrome. In contrast, the population with several mutations that cause the same disease is more likely to have picked up those variants from unrelated people during the group. So what it says, we can easily, well, so how do we know? How do we know that's a founder effect? How do we know that 4,000 people came to Costa Rica? Well, probably because we have this historical or genealogical records. Or we might look at the population and see like, oh, look, this mutation is the same for all these people. And this genetic condition is very common in this population. Well, probably they all, related through maybe 12 generations. So they all descendant of the same people. Now, so that was founder. Bottleneck. Bottleneck occurs when many members of a group die or only a few are left to replenish the numbers. Rebounds in population would contain only those alleles in a small group that survived the catastrophe. Therefore, new population has a much more restricted gene pool than the large ancestral population. Population bottleneck can occur when people or other animals colonize islands as well. So sometimes well, this is there is similarity of the founder effect and bottleneck effect. Usually they say in the bottleneck effect, the rest of the members of population died. And in the founder effect, they just started new colony. But again, the population bottleneck can happen when we colonize islands as well. So this is the example that Lepis people in Micronesia, well, it was a typhoon or disaster, right, that killed most of the people, and only few survived. And the one who survived actually was a ruler, or king, I don't know, whatever you call him. So he was a man in power, and he survived. And he decided that he want to have kids with every probably a woman on that island. And he has this disease called achromatopsia. So it's a blindness when you only see stuff red and white, so no color. So he couldn't see any colors. So and because he had more kids than any other man who was a survival of this typhoon, now alleles for this disease in a higher frequencies in that population compared to the rest of people in other places. Or another example, early human expansion out of Africa. Bottlenecks that occurred in groups that left Africa led to less diverse population as compared to the larger and more genetically diverse populations that remained in Africa. So when we, as a species evolved in Africa, we had high genetic diversity. When then smaller groups left to, I don't know, Asia, Europe, other places. So then we started with less diversity to begin with, right? We started, let's say, 10 people left, and no one of them was tall, so only short people left. So that means we have less in one precise population that was established. Now we have less tall people. So that's kind of like this. So when we look at the people who live in Africa still, they have higher genetic diversity compared to those ancestors, compared to those people who originated from them, right? Who left Africa long time ago, and they now created their own populations. Or some Jew people. So let's so this is the population of people who choose to get married within their own people. And when it doesn't matter if who we are, but if we marry only people who live in a particular area, right, that will contribute to higher incidence of certain disorders compared to people go from Europe to America, from Asia to Indonesia, from I don't know, different places. They go to different places, they get married over there, they have kids. Or if people always marry within a small group of people, then of course the certain genetic conditions will be in the higher frequencies. So here we can see actually this is the these autosomal recessive genetic disorders that are more common in the population if people choose just to marry other people within their own group. So you can see it's pretty high frequency. For example, I saw one over here, like Tay-Sachs disease. It's a horrible disease, and kids die by age of 4. It's 1 in 26. So every baby, 27th baby would have it and die. So that's horrible. That's why we need to travel, go to different places and meet other people. So, and hopefully fall in love with them and build families. So another factor that affect genetic frequency are mutations. Mutations are a major and continual source of genetic variation in the population. It can convert one allele to another. Genetic variety can arise from crossing over and independent assortment during meiosis and numbers of repeats of copy number variants functioning as alleles. So genetic mutation, of course, the major source of our variations in genetic diversity. There is no genetic diversity without mutations. Because if you look at people and you think like, oh, why some people have brown eyes and some blue eyes and some green eyes and so on, it's all, genetic mutations then change our phenotype. The same with our blood type, with everything. So, and we already covered how during meiosis, we can have this crossing over independent assortments that allow all your siblings, it doesn't matter, how many of brothers and sisters you have, everyone is individual, everyone is different, right? Except of identical twins. Natural selection is another factor. No, that's still we're talking about mutation. So natural selection eliminates alleles that adversely affect reproduction. So mean if we have, for example, we have allele, horrible allele that Cystic disease, for example, and kids dies at age of 4. That means they don't reach this reproductive age when they can have their own kids and pass them to new generation. So that's what it says. Natural selection actually removes these alleles from gene pool. However, harmful recessive alleles are maintained in heterozygous and are reintroduced by mutations. But how then this allele for Cystic disease still present? Well, that's because in the heterozygous, one allele is mutated, one is broken allele, but one is normal allele, and the carrier doesn't have a disease. So the carrier can pass it to next generation. So genetic load is the collection of recessive alleles, deleterious alleles in the population. Human behavior and historic events can influence diversity of mutations. For example, PKU. Mutation has a minor impact. Most are silent unless coupled with another effect such as small population size or selection. So genetic load is pretty much, well, if you're looking at a population and see what is the proportion of that allele for disease in that particular group. So here we can see the telltale mutation of PKU when it develops in Yemen, and then people migrated to different area, and that's when we find the same mutation among their children and grandchildren. Now, and the last factor, so I just want to go back. First, I want to see how much we still have. Okay, I don't want to make this lecture too long, but here I just want to go back over to the slide. We're talking about how we can change allele frequencies, and we talk about this non-random mating, migration, genetic-driven mutation. So all of this will change our population. It changes the genetics of our population, and as a result, it changes phenotype, how individuals, animals, plants, human, bacteria, viruses, any living organism, how they look within the population when their genetic pool is changing. right? And the last is natural selection. So we almost done with all these factors. So natural selection is right there. Oh, the previous one. Here. Natural selection is a difference when is the differential survival and reproduction of individuals with a particular genotype and phenotype. So the natural selection, look, tells you that if you have a phenotype, right? If you're a white rabbit and you live in an area where being white make you less noticeable by predators, then you have higher reproductive success. So you will survive, you will live longer, you will reproduce more, and that means you will have more offspring that will inherit your gene. In human example, lactose tolerance and copy number variance, that's when people with some of these disorders either have higher reproductive or low reproductive success. In natural selection, reproductive success isn't important because it is what transmits favorable alleles and beats out the unfavorable ones. This ultimately impacting population structure and therefore driving microevolution. Right, so that means if we have, let's take animal population. If some animals have some advantages that allow them to live longer and reproduce more, then natural selection will favor them, and they will increase their alleles frequencies in the population. Now, there is two types of selection. Negative selection is banishment of a trait because it adversely affects reproduction. And positive selection is retaining an advantageous trait because it enhances reproductive success. So pretty much says like negative selection when animals that are not suited to particular environments, they just die, they don't reproduce. And positive selection when the one that have advantage, they reproduce more. In Darwin's time, natural selection was thought to be primary negative. So Darwin thought like, okay, if you don't suit, you die. Today, the ability to sequence DNA has revealed signatures of positive selection in the human genome. For example, the allele for lactase persistence. Positive selection allows the shepherd to thrive at elevations that makes others sick. So this is example of positive selection. Then people who live in a high elevation, they have some mutations that allow them to be adapted to low oxygen condition. So that's a positive selection that keep that allele instead of removing this allele. Natural selection acts on pre-existing genetic variants, and so it uncontrolled and largely unpredictable. In contrast, artificial selection is controlled breeding to perpetuate individuals with a particular phenotype, example, a crop plant. We have created our pets by controlling their evolution. So natural selection says it's unpredictable, but artificial selection, then we will decide what individuals we allow to reproduce and which one we don't. So, for example, genome sequencing supports a hypothesis of two or original ancestral wolf population in Eastern and Western Eurasia, giving rise to two dog populations. So all our pets, all our dogs are actually a descendant of two wolf population from Eastern and Western part of Europe. Thanks to artificial selection and intense inbreeding, body size in domesticated dogs varies more than it does in any other terrestrial mammals. So that's what says that artificial selection, we created those animals, not because we changed somehow their DNA really, but by allowing only those dogs reproduce that we want to. So for example, if you want your dog to be really small, so you have the puppies, let's say 8 puppies, 9 puppies, some are bigger, some are smaller, but you allow only smaller dogs to reproduce. So you increase this allele frequency for shorter, smaller dog paired with bigger dogs. But here's another example, antibiotic resistance that illustrate natural selection. Antibiotic resistance arises from the interplay between mutation and natural selection. Bacteria become resistant in 2 ways. Mutation pass from one bacterial generation to another by cell division, or groups of resistant genes are passed on transposomes. They are transmitted from cell to cell by plasmids. A particularly dangerous strain of methicillin-resistant Staphylococcus aureus, or MRSA or MRSA, that's how we created bacteria that is resistant to antibiotics. So let's see how it happened. So for example, here we have our Staphylococcus before mutation, right? So that's, they all can be killed by antibiotic. Then in the second generation, one mutation happened, right? You see one red one over here. And just by a chance, well, many mutations happen. This might become blue, this might become like, I don't know, orange, but one bacteria now shown in red, this one is resistant to antibiotics. Now, without antibiotics, what would happen? Maybe it would disappear because it's just one bacteria. But if you now apply antibiotic, you're going to kill all of these that are not resistant. And this one will survive. When this one survives, it will reproduce. So that's a reproduction of selection of antibiotic resistance. And now look how population changes compared to the previous population. Now we have antibiotic resistant bacteria that are more successful. So they will increase their number within the population. That's example how artificial selection, this is unintentional. If we're talking about dogs, yeah, that was our intent to create different dogs. Here, we just by using antibiotics, and antibiotics never kill all the bacteria. Some will survive. Why they will survive? Because by the chance, they already had this mutation. What we did them, we just selected them, right? So we selected those antibiotic resistant and we allowed them to reproduce because we killed everyone else. Okay, so balanced polymorphism. Harmful recessive alleles are replaced by new mutation and persistence in heterozygous. So when mutations are harmful, right, then they can stay in the heterozygous individuals. Recessive condition can be prevalent in the heterozygote if the heterozygote enjoys a health advantage that affects reproduction. So we say that harmful mutations, they are replaced by new mutations. Alleles that lead to a disease and kills individual, they used to be removed. They have tendency to be removed from gene pool. However, what if we have allele that give us a disease, but at the same time, it protects us from another disease? Then it will stay in the population. This is called heterozygote advantage that maintains a recessive disease-causing allele in the population. Balanced polymorphism is a type of balancing selection which more generally refers to maintaining heterozygotes in the population. So just to give you an example, and you probably already heard about this one, we just take one, sickle cell disease. So sickle cell disease, when you have red blood cells that are misshaped, so instead of nice and round, they now the shape of the moon. So this sickle cell disease is pretty dangerous condition. However, it protects people from malaria. from another disease, because atypical red blood cells cannot retain parasites. And malaria, when parasite of malaria, it lives inside red blood cell. So now if we have affected atypical red blood cells, the parasite cannot retain, stay inside blood cells. So that protects from malaria. So that's our balanced polymorphism. So you have one disease, but it protects you from another. That's why it stays in the population, right? So the same with other, like cystic fibrosis, for example. Why we still have the cystic fibrosis? Why wasn't it removed from our gene pool? Well, because it protects us from some diarrheal disease. So that's what balanced polymorphism is. So here, the carriers of sickle cell disease are resistant to malaria or develop very mild cases. Malaria is an infection by parasite Plasmodium falciparum or related species. In sickle cell disease, blood is thicker than normal, hampers the parasite's ability to infect. Bent shape of the cells prevent parasites from growing and block their spreading of infection. Right, so that tells you that even, we have the sickle cell disease, but it's allow you to survive malaria. And another alpha thalassemia and hemoglobin C are two other globin diseases that block the parasite. So again, we keep this alleles in heterozygous population because it gives them reproductive advantage. So you can see that this is the Africa and area where we have sickle cell disease, right? It's actually the same area that highly affected by malaria. So the people, so just imagine people, lived in this area, and malaria killed them. Like malaria still kills 1 million people every year. So people live here and malaria kills them. People live here and malaria kills them. But then mutation happened, sickle cell mutation. And people have the sickle cell mutation. All of a sudden, they stop dying from malaria. Now, guess what? They're going to pass this mutation to their kids. And that's how we have increased this sickle cell allele in the same area where malaria was killing millions of people. Here's a cystic fibrosis that protects us from diarrheal disease that can be major killers. So cystic fibrosis is got like a special SFTR protein that affects chloride channels. Cholera toxin causes chloride channels to open, producing severe dehydration. So, for example, if somebody has disease, cholera, right? That's when you pretty much have so severe diarrhea that you die, right? And this is because this toxin opens this protein, CFTR, and that causes severe dehydration. However, if somebody has cystic fibrosis, then this protein is not functioning protein. Typhoid fever require functional CFTR for bacteria to enter the cell, right? But protection against infections that produce diarrhea may have kept cystic fibrosis in the population. So for you to get this bacteria to have disease and to die from dehydration and from diarrhea, you need a functional protein, CFTR. Now, people with cystic fibrosis, this protein is not functioning. So bacteria cannot enter the cell. That means the person who has cystic fibrosis will not be affected by the bacteria. So you have the chances to die from diarrhea decreases. That's why probably cystic fibrosis is kept in the population. Okay, and this was the last slide. I am sorry, this lecture was a little bit longer than I wanted it to be, but you don't need to watch it the whole lecture in one time. You can just watch a little bit, then pause and watch the rest of it later when you have more time. But anyway, I hope it was helpful, and thank you for watching.